Okay, hi, this is Open Source Antibiotics Series 2, meeting on October 30th. I'll just go ahead and share my screen with the agenda on it. Can you guys see that?
Yep.
Okay, cool. So I guess the first thing is just jump into the new data that we have, which is posted in issue number 35. I think I have pulled up somewhere. Right. So this included some of the DNDi compounds, as well as some of the ones that Ed and I made. So I think it can, yeah. So I guess the question now is, based on this, what should we be targeting next? So does anybody want to jump in or?
So we have some compounds with, so where you've got that exocyclic nitrogen, the NH, we have some compounds. I have to check to see if they would be exact matches back to like with the core scaffold substitution pattern.
Yeah.
But we would have potentially some where we've actually alkylated that nitrogen or replaced it with an ether.
Cool. Yeah, I think those would be worth screening, especially since given the 839 and 851 are active.
Yeah.
Yeah, cool.
So I can double check what we have on campus. We're gonna have the benzylic with the extra CH2, but
this one.
Yeah, we've got a lot now with the sort of 4-fluorophenyl at the top.
Okay.
4-fluorophenyl should probably be okay. You need something in the para position up top, it seems.
Yeah. Yeah, so I let me double check exactly what we have, because we've also, Quillen, who's been working on the project, has taken off Antonio's synthesis. And so they've made a bit of a library now of an alkyl type.
Okay.
So we can check for those.
Let's see. That would be really good.
I have a small PowerPoint of a different couple compounds I spitballed. Just wouldn't mind hearing everyone's opinions on them.
Cool. Go for it.
Let me just get that loaded. I use Open Office, so it's a little finicky compared to Microsoft PowerPoint.
That's fine.
Let's see here. Share screen. You should see Open Office and Press. Open up. Yep. There it is. So let me. You can see all this?
Yeah.
So obviously the first thing we were looking at was we need something in the para position. It seems a little lipophilic is better. Now, that's not great for metabolism, of course, as we all know, because it's most likely to, it's going to probably get shredded first pass. But I was interested in particular in the one with the DNDi type core with the paratoluol substitution. Another one was these N-alkylated ones, which use this other DND provided core. Obviously, you'd have to go through the bromide because methylphenylizocyanate is basically very volatile, and you'd have to go through those cores. But I'm not sure if any of these pique anyone's interest. Obviously, a couple of these are very easy substitutions. Basic style stuff that you'd see in every early drug design program. But,
yeah, so I think Matt also was interested in that DNDi core with the toluol substituent. So I can.
Yeah, the green compound.
Yeah, should be easy to make. I also have
is it worth checking that DNDi haven't got that compound?
Yeah, we could reach out to Ben and see if they have made that.
Yeah, that's not a bad idea. There's also these two. which are DNDI derived again. I think the DNDI core is quite interesting because it seems to be easier to synthesize, if I'm not mistaken, than the one with the smaller ring.
It's a one-pot reaction that we do to make
But this one you can't because the isonitride was unstable.
So we can still make the isocyanides. Yeah, it stinks. Can confirm. Not great. But no, we can still do them. Quillen actually just distills off. Distills off the isocyanide, and so it keeps it all in the fume hood, and that actually. Yeah,
because doing a literature search seemed to result in a lot of. But yeah, this should just be. I mean, this shouldn't be too hard, Just make the.
Yeah, I've ordered the starting material to see if we can make that core through our current route, which
the bromine core, the bromine core.
So we'll see how that chemistry goes. I'm planning to give that a go.
Dana, Antonio, our former visiting grad student, actually did make the core following a similar prep to you, so I can actually share that.
Cool.
He chlorinated instead of brominated, but I would imagine.
Yeah.
I don't think it should be too. I mean, this is very, very well hammered home chemistry in terms of. Another thing I had was this, which was to play around with if there was the way metal binding could. But it's still going to bind in between those two nitrogens, but it might be worth a shot. If it is metal binding, I'm not sure. And this was an attempt to increase water solubility. So this boronic acid is available and it's cheap.
Okay.
So that was all I came up with.
The compounds with the benzo ring. Why have the methoxy there? Do you think it's essential?
It was potent, wasn't it?
Yeah, so there's a match pair in the DNDI compounds where you've got them with and without the methoxy on the core, and the methoxy does add potency.
There's also the methyl group seems to do similar.
Yeah.
So one thing that I would say with the methoxy at that position, what we have found in terms of, so obviously we're looking at a completely different indication in terms of NTDs, but looking at the host cell tox, when you're substituting that 7 position, we do have more toxicity across some of the cell lines than we do if you have the substitution at the 6 position. So moving it up 1 as drawn.
Yeah.
So it might also be, so we have now kind of moved a little bit more away from that 7 methoxy to the 6. But again, I can also have a look and see if we've got any of those, although it's kind of introducing a second variable into the mix.
Did you ever make the compounds with the chloro there?
Off in place of the methoxy?
Yeah.
I would have to double check. If we did anything, it probably would have been the fluoro.
That would be more stable. I was wondering maybe CF3 in that position, but that's going to require a lot of finicking to get that assembled, isn't it?
Yeah, the problem is CF3 is quite lipophilic as well, and we're quite a way up on the log P scale already.
Yeah, I figured as much.
So I also bought the starting material to put a chloro in that position. So I'll give that chemistry a go and see how it is.
I was wondering, have you tried substituting the imidazole or the pyrazole with anything other than a methyl?
No. Wait, where?
The green part of the compounds that are on your slide.
No, the bottom.
Okay. So I guess one of the things that I'm also wondering is whether you could substitute with some sort of, Dana might know where I'm going with this already, saturated cycle like a THP or piperidine?
We have one morpholine compound that was inactive. It was morpholine on the bottom. I don't know. We've not tried
Is
morpholine.
that-
We've not tried.
We haven't done any substituted pyridyls at the southwest portion of the compound.
Not pyridol. I'm thinking like off of the pyrazole or the imidazole NH. Instead of the methyl, switch it out for like something a bit bigger. You could even do something small like the cyclopropyl initially, or the and then kind of work your way out. But it might give you that ability to bring in something like, if you do the piperidine, you would have the NH out there.
I
It's
think
gonna be
it.
quite a
So
bit
yeah. So you're talking about the core then,
Yes, the green part.
right? So we've only made bicyclic cores at this up to now.
Right. Yep.
I've been trying to make the unsubstituted. The chemistry has not been amazing.
I wonder if there's any reports of a tricyclic core of that size.
You mean like a carbazole? Is that what you mean by tricyclic?
Carbazole, maybe something. I don't know. We could try playing with or putting another ring on the edge of the core.
Yeah, that's where I was going with my comment. But
Okay.
I think if you try and do if you try and fuse those rings together, you're going to then be very flat, very rigid, very insoluble would be my guess.
That's right, yeah. I'm not sure that's a direction you'd want to go, actually. I mean, I think if you wanted to put another ring on, you could perhaps annulate it with something like, as you say, a piperidine or a morpholine or something like this, but something that has a heteroatom in it.
Okay. Yeah. Cool. Yes. Okay. I think what I've ordered some chemicals to make essentially some of the compounds that we've already discussed, some of the ones that are really more closely structurally related to ones that we've already made. And then we'll just have to work out synthesis because to do some of these more modified cores, I think.
Yeah.
Cool. Okay. I guess we will move on to next stuff on the agenda, and then we can obviously keep this discussion going on that GitHub issue. Tox assays, they will be done. So Alex, the postdoc, is just checking on her cell line, but hopefully early next week, we'll be able to get the tox assays going. We've picked 8 compounds. That's GitHub issue number 3. So if anybody has strong opinions about that, just go check us look. And then, if we need to change it, we can change it. But I think the ones we have now are a good first step. Okay. Ed, do you want to do the chemistry update?
Mm-hmm.
Okay, cool.
Yeah, mine's just pretty quick. So I've gotten those 3 compounds on the bottom that I was working on. So they should be good for the next round of assays.
Okay, mine's quick too. So the top 2, I've been trying to purify, particularly 147. I've already done 2 columns on, but I do have enough material to try and purify it again. So hopefully I'll get some clean material out of that. 151, I've just done the Bhakti protection with TFA, and it does look to be cleaner. That I haven't done any purification on yet, so I just need to do that. And then this is the procedure that I've used, which I think there's a chance that it has worked. oh, sorry, that should be a trityl group, not a methyl group on the imidazole. They don't ionize well, so I can't really tell by LCMS, but by NMR, it does look like I got the ferronic acid. So I did the Suzuki, and I just have to see if I can isolate the product. So hopefully this will work. And then that should give us, you know, a bit more of an idea of the SAR around the core again.
So you got a trityl on the nitrogen.
Yeah.
It's nearly all protecting group then, really, isn't it?
Yes. Yeah. And just a hydrocarbon too. So
yeah. That sounds hard work.
Yes.
Oil.
Yeah.
So fingers crossed for that.
Yeah.
Okay. I think most of the rest of the stuff on the agenda is on Matt. So I'll just go back to this very quickly.
Well, we can assign him a few more things to do.
The units, I think we sort of resolved that last time, right? And he had more reading to do. Ed will make a video at some point. Newsletter we have sent out. And I don't know if Matt's gotten any responses to that, but hopefully there's some interest in that. And yeah, oh, mechanism of action. So we actually should check in on this soon. I'll just do that, put that on myself, and I'll email Lee because I think it was around now-ish that he said they would expect results. And we're still waiting on Haifa and GSK. Cool. Okay. If nobody has anything else, then I will.
Well, the only other thing I was thinking was that, you know, these structures are quite simple and people may have them on their sample collections. Should we try and do a sort of crowdsourcing to see if we can get anybody else to contribute?
Yeah.
And how would we do that if we did do it?
Yeah. So I think for the MER ligase project, we did try to put out a call basically via Twitter, and didn't get too much of a response. So I don't know if that's the best way to do it.
Yeah.
Yeah. I guess we could.
Unless we do a manual search and email campaign, but that takes time.
Right.
Yeah. I mean, I think it would be a good topic to put in your next email.
Mm-hmm.
Okay. But it would be nice if we had a sort of landing page that you could just point to if you came across anybody who was working in similar sort of compounds or might have them, that we could just point them at.
Yeah. Okay. Yeah, I think that's a good idea. I mean, I guess we could kind of do that through GitHub. Would probably be the easiest way to sort of fold it in.
Yeah.
Okay. Yep. Anybody have any other outstanding business? Okay. I think we probably can leave it there then. And Lori, if you just want to be in touch with me about whatever compounds you guys might have.
Yep, I'm also sending you now the aromatic core that Antonio made. And
okay, I'll just upload that to GitHub so it's available.
Mm-hmm.
Yeah. What was the original indication for DNDi to explore these?
We're looking at it for Chagas and Leash.
It was for Chagas. Okay. Was there ever an MOA done on? Was there ever an MOA study done on trypanosoma?
Not that I'm aware of, no.
Okay.
Okay. All right, cool. And then if anybody has any other, you know, lingering thoughts about what to make next, that obviously can go on GitHub, and then we can also talk about it next week's meeting.
Sounds a
Just
good idea.
coalesce them into a PowerPoint.
Okay, cool. Thanks, guys. I will see you next week.
Right. Thanks very much.
Bye.
Bye, everybody.